Hi, my name is Spencer MacDonald, and this summer I worked on the project Malignant Behavior in Canine Bone Cancer Cells in Dr. Jeffrey Wood's lab in the Pathobiology department. I worked alongside one of his master's students, Jacqueline Gallian, and together we studied primary canine osteosarcoma cells. Osteosarcoma is one of the most common types of bone cancer in dogs, and due to the metastasis to the lungs, carries a particularly grave prognosis. In particular, we were interested in the PRK-AR1A gene and its effect on prognosis of this disease. PRK-AR1A encodes for the R1 alpha regulatory subunit of cyclic AMP-dependent protein kinase A. In the past, it has been shown that in mice and humans, there is a molecular subclass that has a low PRK-AR1A expression and responds better to chemotherapy than a high-expressing osteosarcoma cells. Additionally, I was tasked with a side project to look at autophagy, and I wanted to detect it and investigate if it could have a mechanical process in why this PRK-AR1A has a role in chemosensitivity. Our objectives for this were to knock down the PRK-AR1A expression in primary canine osteosarcoma cell lines, and from there, we would treat them with chemotherapy drugs to compare the chemosensitivity between the controls and the knockdown group. I was also tasked with the autophagy project to determine if these cells could undergo autophagy, and if they did PRK-AR1A have a role in that? Our cells were grown under three control methods, three treatment conditions. The first was the control, which was grown under standard cell culture procedure. The second was the autophagy induction treatment, which we induced autophagy by exposing it to a starvation buffer. Finally, we have the knockdown by siRNA treatment, and they are the same cell lines as the other treatments. We just knocked down the PRK-AR1A expression using siRNA-mediated transfection. We used three cell lines. Two were primary canine osteosarcoma cell lines, the JL31 and the JL75 cell lines. And the D17 cell line was a commercially available osteosarcoma cell line. Initially, we did a Western blot to detect their baseline PRK-AR1A expression. And from there, we had to determine if the cells could be transfectable, the primary canine cells. To do this, we used hyperfect-mediated transfection to transfect with TYE563, which is this red fluorochrome in the JL75 cell line. We also used HeLa as a transfection-positive control. Another method we used was PEI-mediated transfection of a GFP DNA plasmid. With these two methods, it showed that the JL75 cells are indeed transfectable. Additionally, I had my autophagy project, and I initially tried three treatments. I tried the starved without chloroquine, the fed without chloroquine, and the starved with chloroquine. And from this, I got a Western blot, which was unfortunately inconclusive and needed a fourth treatment condition, which was fed with chloroquine. After this poster was made, I completed that experiment, and I did get conclusive results. Unfortunately, I found that I had not successfully induced autophagy. Still to be completed in the future is the successful knockdown of the PRK-AR1A gene, followed by the treatment with chemotherapy, and then subsequent proliferation assays to determine the chemosensitivity of the cells. In conclusion, if we find that PRK-AR1A has an association with chemosensitivity, it can be used as a potential therapeutic target to sensitize the cells to chemotherapy and improve the patient's survival in the long run. I would like to thank the University of Guelph for their URA program for funding my position, and the OVC Pet Trust Fund for funding my research. I would like to thank Dr. Jeffrey Wood for giving me the position, and Jacqueline Gallian and Barbara Jefferson for all their training and support. Thank you.